Stem cells rejuvenate aging hearts. Have you ever noticed how often stem cells are appearing in our news? It's not surprising, given that we're pinning our hopes for a disease-free future on them. But whether it's stem cells from tissue or tissue from stem cells, have you ever wondered, how does it all work? A stem cell is unique in that it can self-renew indefinitely and is undifferentiated, meaning it doesn't have the specific combination of genes turned on or off that will dictate the cell's functions. Every cell in your body originated from a single fertilized egg, or zygote, that is in fact a totipotent stem cell. All of your stem cells have the same genome, but development restricts their fate. Totipotent stem cells can become any cell in the embryo and placenta. Pluripotent stem cells can make up only any of the cells in the embryo. Multipotent stem cells within specific organ tissue can become any of the cells that make up that organ. And finally, unipotent stem cells are limited to becoming cells of one tissue type. But what initiates and controls stem cell renewal and differentiation? Or is it just left to chance? No, it is highly regulated by a large number of biological, biochemical, and biophysical events. While stem cell fate can be controlled internally by the cell, in most cases, it is the result of external influences from the niche microenvironment. Stem cells are found in niches made up of stem cells, other cell types, and an extracellular matrix, or ECM, the scaffolding of proteins and molecules that exist between cells. The niche controls stem cell behavior through chemical signaling between the stem cell and other cells in the niche, and ECM components, especially growth factor proteins and adhesion molecules. ECM pressure and stiffness provide stem cells with signals that influence their fate. Adhesion molecules keep the stem cell in close contact with the niche. Within the niche, the stem cell is kept quiet until the niche receives growth or repair signals. Then, growth factor proteins activate cell division, and targeted niche signals influence how the cell sets up polarity and orients its meiotic spindle before division. A weak polarizing signal orients the spindle parallel to the niche, ensuring equal distribution of cell fate determinants that produces two identical daughter stem cells. A strong signal holds the spindle to the niche, resulting in perpendicular orientation of the spindle and unequal distribution of determinants. Here, one daughter cell receives stem cell determinants and the other is induced to change by non-niche cells. Following cell division, two identical daughter cells may be exposed to different environments that give rise to alternative fates. Although not fully understood, there is a growing consensus that stem cells and stem cell niche interactions may help us realize our dream of curing incurable disease.